Welcome, everybody. We're going to wait just a couple seconds for people to join. Well, thank you all for joining. We are recording this webinar, and we'll have that posted on our website. I'm Mara Sokovich. I'm a neurologist and ALS specialist at Mass General Hospital at the Healey Center and the sponsor of the HEALEY ALS Platform Trial. I'm joined here today from several members of the team that is leading this first platform trial in ALS. Marianne Chase and Lindsay Pothier from our project management team at Mass General. Melanie Quintana from Berry Consultants, who's one of our lead statisticians on the project, and Dr. Sabrina Paganoni, who's the co-principal investigator of the platform trial. So we wanted to use our time to give you an update on the platform trial, tell you about it, and really encourage any of you who have drugs ready to go into a phase 2-3 trial in ALS to come and talk to us and consider joining the platform trial. We have many partnerships. We are running this trial with the Northeast ALS Consortium, and we have many foundations and patient groups that are very behind us and supportive of this approach to try to accelerate how we develop treatments for people with ALS. Next slide, please. So today you'll hear from Dr. Paganoni and Dr. Quintana about the platform trial and the design, from Marianne Chase about the operational structure. And then I'll come back to talk about how you could partner with us if you're interested in joining the platform trial. And hopefully we'll have time for questions. Next slide. I'll pass it to Dr. Paganoni.
Yeah, thank you so much. Next slide, please. And welcome everyone. So I want to start just by sharing why we're doing all of this. One of our patient advisors, Sandy, taught us that patients with ALS do not live on the human time clock because the ALS clock is so much faster. So we really wanted to build a trial for ALS patients that would address that urgent need to develop new treatments faster. And the opportunity came in 2018 when the Healey Center was founded. And actually it was Dr. Sokovich who had the idea of bringing the platform trial approach to ALS for the first time. Because our research had shown at the time that if we were to adopt a new trial design, we could dramatically accelerate the time it takes to develop new drugs. So our research showed that, learning from oncology, that a platform trial would cut the drug development timelines in half, cut costs by a third, and drastically reduce the need for placebo. So we really wanted to do that, to do something that would really fill that gap. The platform approach does require a great deal of planning and collaboration. So I just want to go to the next slide and share that at the heart of the trial lies the Trial Design Committee, which is a dedicated committee of ALS investigators from both the Healey Center and the NIS Consortium sites, as well as statisticians from both the MGH Biostatistics Center that has extensive experience with ALS trials, and Berry consultants who are the world's experts in platform trials. And we work closely with our operations team, and you'll hear about it later. Next slide. We also worked with the entire ALS community to launch the trial rapidly and efficiently. And actually it was a challenge to launch it right before the pandemic, but we're really seeing the fruits of that, as we'll discuss later, because really we came together as a community to launch something that would fit the needs of not only patients, but also industry, regulator, and the clinical trial community. And we had a series of meetings with industry representatives at the time in 2018, 2019. We wanted to include their perspectives in the design. And they told us that basically they wanted to have a trial that would provide them access to experts in ALS science, that would be well executed with high quality and solid infrastructure, and that would be efficient and kind of agreed upon with the FDA so that we could move new drugs forward in an efficient way. And so that's exactly what we did. And so we engaged with the FDA from the very beginning, and we're really grateful for their advice, especially Dr. Billy Dunn and the entire neurology division. So we had early and frequent communication that really informed the design and the statistical analysis that you'll hear about from Dr. Quintana. And we also continue to engage with NILS trials, NILS sites. And so we have so many sites that are engaged and really excited to put their patients into the trial. So I think that really, the initial collaboration really was key for the launch and success of the trial. Next slide. So in basic terms, the way this works is that rather than running each trial or testing of each drug individually as a standalone trial, and therefore building an infrastructure for one trial at the time, we built an optimized infrastructure that leveraged input from all stakeholders at the beginning. That was 2019 or so. And then when we launched, we had this great open infrastructure that could accommodate many drugs. So now we're testing multiple drugs at the same time. We can continue to add drugs. And that's where we gain those efficiencies in terms of time, costs, and again, overall patient-centered approach. Next slide. So the backbone of the trial is the so-called master protocol, which is available on our website. It's a common protocol for multiple regimens. And so the master protocol is like our house, and it has strong foundations that include our target population, the endpoints, the sample size, and all the features that are important to any trial, but also some unique features that are important to a platform trial, such as placebo sharing and adaptive features. So our house can accommodate many guests, and so each guest is one investigational product. We call that a regimen. Next slide. So each regimen has its own room in the house and is described in a dedicated regimen-specific appendix, which is actually an addendum to the master protocol. So every time we add a new regimen, we don't need to have a new protocol approved. We just have to have the addendum approved. And so we started with 3 regimens, A, B, and C. Then we added D. Then we added E, and now we are continuing to add more, and that's why we're having this conversation. Our goal is to always have spots available for patients so that by the time we're done enrolling for the first few regimens, then we are ready to go with the next one, so that we always have patients waiting, always have patients wanting to enroll. And that's why we want to continue to expand our house. Next slide. And so from a patient perspective, I just want to show how the workflow is from the patient perspective. So we have a 2-step informed consent and randomization. When a participant enrolls in the trial, they're randomly assigned to any of the available regimen. And this first assignment is random, but it's not blind. So we will know which regimen they're assigned to. So for example, one regimen could be assigned to regimen A, and the participant will know that potentially, they'll get, they're getting either active or placebo within regimen A. Then there is a second level of consent and randomization. And this second level is actually double blind. So the participant will be randomized to either active or placebo within that regimen. So for example, participants assigned to regimen A, they'll know that they are in that regimen, but they don't know if they're getting active or placebo. Now, because we have this common master protocol-based infrastructure, we can share placebo data among regimens. So the randomization ratio within each regimen is 3-to-1 in favor of active. So there's many more chances of being assigned to active rather than placebo, which is great from a participant perspective. But then from an industry perspective, at the end of the trial, each investigational product is actually compared to the shared placebo group with a active to placebo ratio of 1-to-1. Again, you gain efficiencies both from a participant perspective and from a company perspective. Next slide. So in each regimen, we are enrolling about 160 participants, 120 on active and 40 on placebo. But we need to remember that the active data is then compared to the shared placebo data. So at the minimum, we have 240 individuals for each regimen, 120 on active, and then 120 on placebo, including shared placebo data from other regimens. Now that we have so many regimens that have already completed or are ongoing, we have even more placebo data to leverage. The double-blind portion lasts 6 months, and then at the end there is an open-label extension that's very important to capture long-term safety and efficacy data. Next slide. So we put a lot of thought into our target population, lots of statistical simulations that Dr. Quintana will explain. And the key principle here in the inclusion criteria is to allow access while also maintaining good statistical power. We do allow standard of care medications, and we certify participants by their use in alignment with FDA guidance. Next slide. So our primary endpoint is the change in disease severity through 24 weeks as measured by the ALS Functional Rating Scale. And the design is such that the trial has the potential to provide confirmatory evidence with an overall type 1 error of 5%. If you can click one more time, I want to show the secondary endpoints that are customary for ALS trials. Click one more time. I do want to point out that we also have an extensive set of exploratory endpoints, which are very important because we want to take advantage of this trial to also contribute to the field. Next slide. So for all patients that are enrolled, we do deep genotyping, deep phenotyping, extensive biofluid collection, because we want to test emerging biomarkers such as neurofilament levels in all participants. We want to leverage these biosamples to test new biomarkers, and also we can use them to test drug-specific biomarkers that are applicable to specific investigational products. We're also developing novel digital signatures of speech production and respiratory function. And again, with all that's going on in trials, I think that it's really important that we continue to develop these new tools. And then as we learn from them, from this growing infrastructure, then we can adapt future regimens and also inform other trials in terms of more efficient measures for ALS trials in general. Next slide. So the level of interest from sites and patients has really been phenomenal, really amazing. During the pandemic, we never stopped. We really continued to enroll at great rates. We selected initially a group of about 50 sites, and then many more wanted to participate. So at this point, we have a network of 74 sites. Next slide. My favorite aspect of this is that we can connect with people living with ALS on a daily basis. And in fact, just before this webinar, we had our weekly patient webinar. Every week we are online, and with our patient navigators, we have a dedicated team of people who are in contact with our patients on a daily basis, but we also meet as a group and host these weekly free webinars. We really have been connecting with thousands of people with ALS, you know, from all over the country and really from all over the world. We also have weekly newsletters and lots of opportunities for communication. And I think that this really helped us with enrollment. Next slide. And again, this is a perpetual adaptive trial. We started with a few regimens. At this time, we have 5 regimens that are enrolling. Many people, hundreds of people in the trial, hundreds of people also on open label extension. And we expect the results of the first 4 in 2022. Next slide. And we published our first paper. We have more papers that we are working on. The first paper was published in Annals of Neurology to describe the design of the trial, and our master protocol is available on our website. So the link is on the slide. And with that, I think I will turn it over to Dr. Quintana.
All right, thank you. So I am going to speak on behalf of our great, our statistical dream team that we have here that we've been working with, myself, Dr. Macklin, Dr. Seville, Dr. Bind, and these are just the blinded statisticians at this point. There was a larger team when we started designing this trial, but several of them had to move over to the unblinded statistical analysis committee at some point. Next slide. As Sabrina mentioned, the primary endpoint is the change in disease severity through 24 weeks. And we measure that with the ALS FRSR as well as mortality. And I'll get more into that in a little bit. The trial was designed to have the potential to provide confirmatory evidence towards an NDA with an overall type 1 error of 5%. And as Sabrina mentioned, we have 3-to-1 randomization for each therapy, that is active treatment versus placebo. And we share placebos across all what we call regimens. Additionally, we use concurrently randomized and historical sort of non-concurrent placebos as well. So the placebos that might be a part of regimen F, we'll still be using those who were a part of A, B, and C. The platform trial is also adaptive, in that we want to efficiently find therapies that work. And there are frequent platform-wide interims to allow stopping regimens for early success or futility. Right now, built in, we have for the first several regimens, just the futility stopping, and I'll go into that in a second. Next slide. The master protocol itself, we think of as kind of the global rules that everyone follows by. We have trial eligibility, visit schedule, randomization ratio of 3:1, study duration, primary endpoint. And then we have some recommendations in terms of sample size per regimen, what primary analysis to use, what early success or futility criteria to use. However, each regimen, when they come in, will create their own regimen-specific appendix. And there'll be some customizations based on the need of the sponsor. So there may be additional restrictions on inclusion or exclusion, but only due to safety or mechanism of action. We want to make sure that we're enrolling similar patients across the different regimens so that we can share the placebos across them. Additional endpoints may be collected, and a sponsor may have different study stage goals. So the success threshold might be different for a purely phase 2 study than one that you want to potentially use to provide confirmatory evidence. The primary analysis may be changed. There might be a bigger or smaller sample size. There might be more aggressive futility. So really, once a regimen signs on, we customize to fit their needs and create this regimen-specific appendix. Next slide. This is just a schematic of the trial, and I think was already explained very nicely, as that from the patient perspective, there's sort of this 2-part randomization. First, randomized to a regimen, and then within that regimen, randomized 3:1 active to placebo. Now, what that means is every comparison is going to be the treated. So say we're talking about regimen D, we'll look at those treated with regimen D within regimen D versus the entire shared control. And as I mentioned, this may be concurrently randomized or previously randomized controls. This may mean controls from other regimens that had a different mode of administration, or controls from other regimens that had different minor inclusion exclusion. And we stress the word minor because, again, we want to maintain the comparability of these controls across the different regimens. Next slide. Again, I mentioned this platform is adaptive. The interims occur every 12 weeks in the platform, and these go on until the platform ends and we find a cure for ALS. A regimen would be active in a interim analysis if they had some minimum amount of data. So right now we define that as 40 participants with at least 24 weeks of follow-up. And at each interim, right now, the regimens just have futility stopping, and they would stop early for futility if the probability that we slow disease progression by at least 10% is less than 5%. And I'll go into the operating characteristics of this choice, but it's really quite conservative in that we only end up stopping regimens whose where the treatment group is progressing faster than the control group. So your treatment effect would be in the opposite direction as you would want it to. And there is the potential in these interims to stop early for success as well, although we haven't built that into a regimen just yet. Next slide. Now I want to talk a little bit about the primary statistical analysis for each regimen. As most of you, I'm sure, are aware, within ALS, the FDA guidance states that mortality should be integrated into the primary outcome by an analysis method that combines survival and function. So within ALS, that's our main goal, is to have a primary analysis method that combines survival and function. However, within the platform trial, we also have some unique complexities that aren't there for normal traditional standalone trials. And that is that we have these controls that we are sharing information on, and we need to be able to account for potential differences in the controls, either over time due to being concurrently randomized versus not, or potential differences due to, say, mode of administration or different minor inclusion-exclusion criteria. So we have these additional complexities on top of needing an analysis method that combines survival and function. Next slide. So our primary analysis is what is a Bayesian shared parameter analysis of function and mortality. The ALS-FRSR, the functional component, is a very traditional repeated measures model with a linear rate of progression and measures the slowing in that rate of progression. You can kind of see that in the plot on the upper right-hand corner. And the mortality component is a traditional exponential time to event, again, with this slowing in disease progression, similar to a hazard ratio. What combines these two components is this common slowing in disease progression that we estimate between the two of them. We call this the disease rate ratio, or you can think of this as 1 minus the % slowing in the rate of progression. Now, the treatment effects in mortality and in function may be different, and that's okay. This overall estimate provides a weighted average of the treatment effect in the two components, weighted based on how much data is in each. So if all the patients experience mortality and we have very high mortality rates, then most of the treatment effect would be estimated by how well the treatment can slow mortality and increase survival. However, we don't think that this will be the case. Likely in our patient population, there'll just be about 5% participants who experience mortality, we estimate. And so the treatment effect will really be mostly estimated by that potential slowing in function that you see in the top right plot there. Because we're in the Bayesian setting, the Bayesian statistics really nicely allows for evidence or data to be synthesized across different sets or different sources of information, such as across different regimens. And so we're using this Bayesian setting so that we can adjust for the differences in the shared controls over time and across regimens. And I'll just also state that we worked very closely with the FDA in designing this. And I think at this point, they understand this analysis and have given the green light on this analysis. That being said, next slide. With this analysis, you do need to justify any assumptions it makes and perform sensitivity analyses around those assumptions. So with any sort of complex, any analysis in general, you really want to list all of the assumptions, characterize how the model performs based on those assumptions, and then have this set of sensitivity analyses. The main set of assumptions in our primary analysis model is that linearity over time. And so we'll do a sensitivity analysis around that. That common proportional slowing in disease progression, that the treatment effect is this multiplicative slowing in the rate of progression. And we do a sensitivity analysis around that to say, okay, maybe the treatment effect is different over time. And then this common treatment effect between mortality and function is another main assumption. And again, we do a sensitivity to estimate the two separate components. And then finally, the sharing of similar controls. We do different sensitivities where we look at only regimen-specific controls or only concurrently randomized controls. And we have this long list of sensitivity analyses. And what we expect to see is that there's consistency, although we don't expect that we'll be statistically significant on all these. We want to see consistent results across all of these sensitivity analyses. And finally, the FDA wants to see that traditional CAFS analysis or joint rank analysis that will be performed as a key secondary analysis, and they'll want consistent results on this one as well. Next slide. So quickly, I'll just go into how we designed the platform trial. How do we know that we have good operating characteristics under this sort of more complex, innovative primary analysis, under the different futility stopping rules or potentially success stopping rules? How do we know our sample size is big enough or the length of follow-up is enough? Well, we do clinical trial simulation, and clinical trial simulation helps us understand operating characteristics of the proposed design. It helps us optimize the design itself. So we did lots of simulation when we were in the design phase. And it helps us quantify efficiencies of the proposed platform trial over a more traditional approach. Finally, it also, and I don't have listed here, allows us to just understand a more complex, potentially primary analysis method. And these were really invaluable when speaking with different parties, such as regulatory agencies and industry partners, to get them to understand how the primary analysis model works. These clinical trial simulations, the backbone of them is the Prote database. We have this rich, beautiful Prote database that has over 23 historic ALS trials and ALS FRSR data from many individuals over a long period of time. So we use this Prote database to create very realistic virtual patient simulations, realistic data that might come into the trial. And then we perform the adaptive design or the primary analysis method, and we do this thousands of times to see on average how we expect it to work. Next slide. And here's just a picture of that PROACT database. I won't go into too many details here, but again, extremely rich database. We can restrict the data to those who would meet our same inclusion-exclusion criteria. This is what some of that data looks like. These gray lines are sort of ALS FRSR trajectories over time. The red line is sort of the average decline over time. Next slide. And as I mentioned, what these simulations allow us to do is to understand the proposed design. So the one thing that it allows us to understand, if you look on the right, is how much power can we gain by sharing controls. And this shows power just with, say, the 40 shared controls from a single regimen, all the way up to sharing controls from 2 other regimens. And of course, we have even more than that in the platform trial now. So that takes us from less than 60% power to over 80% power by sharing these controls. This is really the invaluable resource that we have in the platform trial. And it's why we want to make sure we can protect it and be able to share these controls across regimens. Some of these others is that it shows that we're powered for about a 30 to 35% slowing in disease progression. We have about 77 to 88% power. And the top row kind of shows our type 1 error. This is a one-sided 2.4, approximately 2.5%. But these are some of the operating characteristics that you would see, especially if you wanted to look at some customizations of sample size and different things within your regimen. Next slide. Might be taking a little longer because I think we have a lot of data in that slide. Yes, so this plot kind of allows you to understand the futility role. And this is what we showed a lot when we were looking at different futility roles and what sort of trials would stop early for futility. What would they look like? On the right-hand side, we have 3 potential interim times, about 7 months after startup, we estimated, and then based on accrual, then 10 months, 13 months, and then 16 months into the trial. The first 3 are early interim analyses. And the dots there, the red dot meant this trial would stop early for futility. A purple dot, which you see very, very few of, is a trial that could potentially stop early for futility, but may have gone on to be successful. And this is what we really want to make sure we limit. And that was less than 1% of the trials are those purple dots. What you see here is that a trial wouldn't stop at the first interim unless they had like a 20% increase in the rate of progression. That's what this DRR of 1.2 means. At the second interim, it would still have to be about a 10% increase in the rate of progression. And at the third interim, it would be about a 5% increase in the rate of progression to stop early for futility. So this is really a conservative rule. We have it in place just so that if a treatment is really not looking to be effective, we can move on to the next promising therapy. Next slide. Okay, and now I will pop over and allow Marianne Chase to go into the operational structure.
Thanks, Melanie. Welcome, everyone. So if you want to go to the next slide, Jen. So I'm speaking on behalf of a very large operational team that oversees the platform trial. We have about 40 staff members internally. So that's in project management. We also work on site management, our team that builds our electronic data capture systems, our data management group. We have a quality assurance and regulatory team, as well as an administrative group that also manages grants and contracts. We additionally have obviously some very key vendors that we work closely with. So I'm just going to give a broad strokes overview of how our operations work for the platform trial, just to give you a sense of that. So next slide. So this is the overall governance of the platform trial, with the Healey Executive Committee sort of being that central team. There are many different groups that report into them, and we'll talk a little bit more about these. I'm going to talk about the Platform Trial Design Committee that both Melanie and Sabrina had mentioned, and we have an operations team. I'll talk a little bit about that. Merritt's going to talk about the Therapy Evaluation Committee. We have some task forces that we form as well. As Sabrina said, we have a fantastic patient advisory group and have very close contact with patients. We have another foundations and benefactors team. But what I'm going to focus more on a little bit is really the regimen steering committees and how we operationally run regimens. And I'll also touch on safety and how we manage safety. And we have a separate data and safety monitoring group. They oversee the safety and all of the regimens for the platform trial. So we do that at a platform level. For each of the regimens that are run, we do have a steering committee, and an industry representative would be a part of that regimen steering committee. And those committees typically meet quarterly. Additionally, we have an operations team set aside. Both we have it at the platform level, which is the large group, and then we set aside time for each individual regimen to also have an operations team, and they meet on a regular basis as well. Next slide. So the design team, which is really where we start, we start developing what has already been described as the regimen-specific appendix. So again, we have our platform trial that really lays the groundwork for our protocol. Each individual compound that comes into the platform trial develops their own regimen-specific appendix. So we set up design meetings where we talk with the clinicians and the statisticians about all of the different pieces that need to come into your particular regimen that are specific for your investigational product. We do have frequent meetings with the FDA, as we've already talked about. The FDA has told us that for every compound coming into the platform, if there are questions to be asked, they will entertain us to have a Type C meeting for each drug and to ask any questions that are pertinent before moving forward to adding it to the platform. And we've done that with most of our regimens. At these design meetings, again, we further define the patient population. So as Melanie had talked about, we try to keep the patient population very similar, but there is customization so that if there are specific safety parameters or things specific to your investigational product where we need to modify that inclusion-exclusion criteria, we define that there. We also look at other pieces of that regimen-specific appendix and customize that for each individual drug that comes into the platform. In addition to that, we have operational team meetings. And at these meetings, we really talk about, coordinating all of the regulatory submissions and all of the pieces that need to go to adding a drug to our platform trial. We talk about drug supply. We have an outside vendor that does all of our central pharmacy. We start talking about developing case report forms, and you can already see, there's a lot of efficiency because we're doing a platform trial. We have our standard set of case report forms for the platform trial. And then we just need to customize those that are different for each regimen. We develop the database, we develop all the different plans and manuals. Again, we have platform level plans and manuals already in place for the platform trial, but with each regimen, we do customize each specific plan. And they have really operational aspects about the regimen specific appendix in those. So we already have those in place. So next slide. One of the key things when we were setting this up is working with clinicaltrials.gov. There were not a lot of platform trials that had been posted on clinicaltrials.gov when we started, and we worked very closely with the leaders there. We do set this up in a unique way. So I just wanted to touch on that here, and I'll talk a little bit about some of the other regulatory aspects. So for clinicaltrials.gov, and you can check this out, it's on there. We have one posting for our master protocol, and it lists all the main characteristics of the master protocol. Then for each regimen that gets added, we add a new clinicaltrials.gov posting. So it has its own NCT number, and we list that regimen. And that way it allows for, at the end of each regimen, for results to be posted. And that was really important for the community and for clinicaltrials.gov to allow posting of the regimen results when they're completed. Because as you know, this is a perpetual trial, as already been talked about. The trial, the master protocol doesn't end, but each regimen certainly will have an end to it. Next slide, please. So now I'm going to just give a really fast overview of sort of the regulatory communication. So just to start with, MGH, or more specifically, Dr. Skovic, is the IND holder for the ALS platform trial. That means we do all of the submissions to FDA from Mass General. So if you can just click one more time. So we work very closely with each one of our industry partners, and we would work with you to understand your drug. So get your investigator brochure, obviously be provided with any updates to that investigator brochure. As we add you to our protocol and our FDA submission, we would need your letter of cross-reference, and we work very closely with you to get all of that information. Click one more time, right? We, as I said, we do all the submissions to FDA. So we start off with our meeting requests. We submit your, each one of our drugs that get added, or our regimens get added to our IND, which is already open, as an amendment to that IND. As we add each amendment, each new drug, FDA has requested that we provide them with a full 30-day review period for them to provide feedback. And then we move on to submitting you to our IRB, which we use a central IRB for this platform trial. We also submit all expedited safety reports, and we do an IND annual report. So those are all submissions that we do. And if you can click, I think one more time, Jen, we also recognize that you as an industry partner will also have obligations to the regulatory agencies. So we provide you with data that's required for that. So for you to be doing expedited safety reporting, as well as your annual DSUR, we would be providing you with that information and that data so that you could then also submit those to your IND, your open IND. Next slide. And safety reporting is obviously very important in oversight. So just to give you a sense of this, we work with a vendor for this. We have a medical monitoring team and a DSMB that, again, oversee things at a platform level. Our medical monitors review SAEs in real time. They also review adverse events, labs, and they do medical coding for us on a monthly basis. Our industry partners, we ask you to always identify a medical representative, and that person participates in the evaluation of every SAE that's reported for your individual regimen. And you're also provided with a copy of our monthly medical monitoring report, and typically you would meet with our medical monitors just to review overall safety on a monthly basis. We have as I said, with many of our plans, we have a platform level safety management plan. And then in addition to that, we would review that with you as you joined the platform. And then in addition to that, we would develop a regimen specific plan for each one of the investigational products that come into the platform trial. Next slide. I think that was the brief overview of operations. So I'll hand that back over to Merit.
Thank you. Thank you, team. So hopefully we'll have time for some questions. Just next slide. I want to say that we are looking for a couple more companies to work with for late 2022, early 2023. So we just want to encourage anyone who might be about 6 to 8 months away from wanting to start, or maybe even less, to reach out to us. We have a therapy evaluation committee. The members are here. It's chaired by Dr. Tim Miller from WashU. And we ask people interested in joining the platform trial to fill out a pretty easy form that's on our website. It's really about the mechanism of action of your drug, what the preclinical science is, and some of the pharmacokinetics of the drug, and are you really ready for that phase 2-3 type? Like in particular, do you have enough information about your dose? And then if someone has all that, then we get to call you up and say that we want to work with you. If maybe there's more work that needs to be done, for example, you need another additional dose finding studies or of a phase 1, we might give that feedback. But our goal really is to try to work with all of you and bring the best treatments forward in the platform trial. We meet every 2 months. We can meet ad hoc. We've received 35 applications to date. And again, we'd like to add about 2 or 3 every year. We could do more, obviously. There have been some platform trials that had 14 arms, but the reality is we think having 3 or 4 at one time is a good number. Next slide, please. So if you can do the fancy new thing where everybody does the QR code and download the application form here. It's also found on our website. As you heard, we are now enrolling in the 5th regimen. We're working with 2 other industry partners on the design, and we are accepting applications anytime for new investigational products that are interested starting late 2022 or early 2023. And I think that might be the last slide. Sorry. Yeah, and now if people have questions, I think you probably have to put them in the Q&A or in the chat. I'm not sure if we can unmute people. So please ask anything that's on your mind and we can read out that question. We're really bummed that we're not in person. When before we started this whole platform trial, we had an industry roundtable at one of the local hotels in Cambridge. We had about 25 companies, and that was really fun to really have a good dialogue. And we do hope to do that again, maybe this summer, depending on how the wastewater COVID levels go. Hopefully it'll stay low. So if somebody does want to speak, we can unmute you. So there's a question. Thank you so much for the overview. How many sites are currently active? How many sites would you expect to be active by the end of the year? Sabrina, do you want to answer that one?
Yes, so we have, I had to say, I had to make the calculation in my mind, but basically we had 54 sites active for the first 4 regimens. They're still active. And to those, we added a few new sites for Regimen E. So when we say active, again, we have sites that are enrolling for E, but we also have many more sites that are active in the platform, still following patients on open label extension, etc. The entire network is 74 sites. And again, between A, B, C, D, and E right now, I would say it's maybe about 60, Marianne.
Yeah.
Something like that. Yeah.
Yeah.
Yeah. There's a question about whether we have sites in China. I'll say that right now, the platform trial is all US based. There have been platform trials that have been international, and we just haven't gone there yet, mainly because all the advice we got from other groups doing platform trial was to start simple and in one country. So that's what we did. So right now we don't have any sites in Asia. We have a lot of collaborators in Asia on ALS. There's a question if we could discuss typical recruitment timelines for Regimen and the average dropout rates. So I'll say the average dropout rates we probably won't share now because we're still like active in the first regimens, but we can say what our assumptions were, and we're less than our assumptions. And Melanie, I don't know if you remember our assumptions for the when we did the sample size of what the dropout rate would be.
So we expected 5% mortality over 24 weeks, and then non-mortality dropout, I believe it was 2 to 3% per month. But I can, and I know that we're less than that. Yep.
Yeah. For enrollment, it varied a lot throughout the pandemic because of the pandemic. There were times when we had sites enrolling 2 to 3 per site per month, and other times when we had sites enrolling 1/2 to 1, and a lot of it was related to what was going on in the pandemic. So it's hard to say. As that, right now, I think we're in a good time in terms of the pandemic. With an IV drug, it's a little bit more than 1 patient per site per month, but we're kind of in that ramp up stage for Regimen E. So there's a question, maybe, I don't know, Sabrina or Marianne, how long is the average time from submission to Healey platform to enrollment initiation?
Sabrina, I'll jump in. So I don't know about submission because I think that goes to the therapy evaluation committee. So I think that's depends on when you submit and when the committee meets and that gets determined. But I will say from when we start design, we typically say design is about 3 months. It depends, again, if there's going to be a type C meeting, we need to schedule that in there. And obviously there's a regulatory timeline for that. But we do try to work to get your regimen-specific appendix developed over a 3 to 4 month period, and we start on operations from there. So I would say typically about 6 to 8 months. Sabrina, does that sound about right to you?
Right, it considering from the very beginning, right?
Yeah.
There's a question about if we're concerned about the assumption of ALS FRS linearity after the AMWITS Advisory Committee. I'll say no, I'm not, but I'm not a statistician, so I'll have Melanie answer.
That was one of the first questions the FDA asked us to justify the linearity assumption. I'll say the linearity assumption is population dependent. So it depends on who you're planning on enrolling. And what we did was justify that linearity assumption using thousands of patients in PROACT that met the same inclusion exclusion criteria, and that seemed to suffice for the FDA. That we do believe it's actually strikingly linear within this patient population in the PROACT database. On top of that, the sensitivity analysis that we have planned is exactly what the FDA recommends, sort of a piecewise linear, less parametric approach. So no, I agree with Merritt.
Yeah. And while we're waiting for any other questions, I'll say, so not all of the Regimen partners that we've are working with have chosen the option to have a Type C meeting. So it's really something we discuss in the design. If there are questions, the FDA has been very supportive of the platform trial and has really said that we can have as many as we need. So they're available, but if a company doesn't have any questions, you know everything about your drug and your dose, you really might not need to do it, in which case then we can move it even faster. I have to say that the IRB efficiencies are pretty enormous. So because the IRB, as you heard, is only reviewing what's unique about the new regimen we're adding. They're not reviewing the platform, the statistics, or the whole setup or the operations. It can be very fast. It's been as fast as 9 days, and we are using a central IRB, and that is just for those of us on the call who've been doing trials a long time, it's really quite efficient. I don't see any other questions. Or for the panel, are there any other points that you want to make?
I think the only other one, just to go along with your IRB comment, Art, would be about contracting. Again, I think that typically contracting in clinical trials has been one of the things that holds things up. But again, because we have a contract with all of our sites and we just add these individual regimens as what we call a task order, again, that speeds up the process for site activation as well.
Yeah, I would just say that from a site perspective, from an investigator perspective, this trial has really changed the way sites think about trials for their ALS patients, because now we know that we have slots available for our patients essentially at all times with new regimens coming in. So I think it really has made these more kind of a high throughput operation. And so really patients are really excited to participate. And so really, I think this is really something that, will continue to grow because of that great patient demand and also engagement from the sites.
Right, I don't see any other questions. So if we will have this recorded and up on our website. I think most people on here, hopefully all of you know how to reach us. Please reach out with any questions, and hopefully we'll be talking to you soon about your investigational products. Thank you.
Thank you. Thank you so much.
Thank you, everybody on the panel.
Thanks, everyone. Thanks. Bye.